Hello, everyone. I am Nadia Rahman Sylvia and have been working with Dr. Stephen Backus for the last 2.5 years. So my research is on autophagy. So what is autophagy? Autophagy means self-eating. It is the cellular degradation process in which cellular contents are encapsulated by a double membraned vesicles, as you can see right here. It's called autophagosomes. And it is delivered to the vacuole to be degraded and recycled. So there are many cellular components that works as machines that helps our cells to function. So under nutrient depletion condition, non-selective autophagy is triggered and it functions as a recycling process of our cell where the vacuole is the recycling center and the autophagosomes are garbage bags that collect all the degraded machines, in other words, cellular components, and takes it to the cell vacuole where it is recycled and reused for cell survival. So why is this autophagic process important? It is important because when the autophagic process fails, diseases like Parkinson's, Alzheimer's, cancer, and all those fatal diseases occurs. So to be able to find cure to those diseases, it is important to first find how this autophagic process works. So there are about 32 different autophagic proteins that helps the process of autophagy, where we know that ATG7 affects the size and the number of autophagosomes. It activates ATG10, and then ATG10 attaches ATG12 to ATG5 and 16 to form this complex that causes ATG8 to attach to lipid PE, which is on the autophagosome size. So it affects the size of the autophagosome. So what I am interested in is the ATG10 protein, this middleman here, and how this ATG10 protein affects the autophagic activity. This is the structure of ATG10. And now let's talk about what we have done so far in our lab. So we have created ATG10 knockout mutants by removing the ATG10 gene from the yeast strains by homologous recombination. We have created ATG10 mutants by site-directed mutagenesis. We have verified our mutants by PCR techniques. And then we have performed Western blots to measure the downstream protein conjugation of ATG8PE at APE1 maturation to look into the autophagic activity. So what is APE1 maturation? APE1 protein is a protein that is present in our cell that is once taken into the recycling center, it activates and matures. So this kind of shows that autophagic activity has occurred. To be able to see our ATG10 proteins under Western blot, we have tagged the C terminus with PA. And as you can see here that our PA tagged ATG10 is fully functional as it shows the same activity as the wild type under starvation condition. We have further expressed our mutants and ATG10PA into ATG10 knockout yeast strains. And as you can see here that all of them have expressed. The tyrosine to glutamine mutant has expressed a lot more than histidine to alanine and the wild type strains. But this is normal and it is confirmed by the Inagaki lab that has published this mutant expression before. And as you can see here that tyrosine to glutamine expresses a lot more than histidine to alanine. We have further expressed our mutants and ATG10PA into the ATG10 knockout, ATG12 knockout yeast strains, and it has expressed well when it was treated with PAP antibodies. So what's our next step? Our next step is to transform HA ATG12 into the strains and look into the ATG12, ATG5 conjugation that is caused by the ATG10. Then we will be measuring the ATG10-8 PE conjugation and APE1 maturation to measure the autophagic activity and to see the partial loss in autophagic activity by our mutants via Western blot. We will be doing alkaline phosphatase assay to further verify those mutants with partial loss in autophagic activity. And then finally, we will be doing transmission electron microscopy to see the effect of ATG10 mutants on the autophagosome size. And that's all. Thank you so much for listening to my presentation.